So, good afternoon. Just shut off the Beastie Boys. It's time to get to Bio 212 test number 3 review, where what we're doing is we're covering the immune system, respiratory, and I didn't spell that right, did I? And digestive systems. Let's fix that to what it should be. Right. Oh. I'm not exactly gaining confidence. Respiratory. There we go. Oh, no, we didn't. My heavens. There we go. Oh, look, did I get my faith back? Did you get your faith back in me? But anyway, let's get to it. Let's get to the first question. What is the lymphatic system? What makes up the lymphatic system? Are there larger structures that are part of the lymphatic system? What types of cells will you find in the lymphatic system? So this is obviously a multi-part question that you can begin by, I would say, first organizing your thoughts around what is it to survey the human body, right? I'm talking about lymphatic system first, so I'm going to draw a body. And I found that if I go slowly, believe it or not, there are fewer mistakes that happen with said pen. So this is not going to be completely anatomically correct, but good enough. Okay? And what I want people to understand is Mother Nature now needs to figure out a way to survey what's inside the body. And what I mean by that is she's going to put a series of lymph nodes going to be attached to ducts, and those ducts are going to attach to trunks, and we're going to be able to collect all of the fluids, interstitial fluids, into specific areas. The area with the lowest pressure, of course, is that cisterna chyli, that then ascends to return materials to your vasculature to then return it to the circulatory system. But here's what I want to make sure you understand. So let's draw this first, right? Put this thing, because we already have the color green in place, right? And let's now move over here. And I know you guys understand the basic idea. You've already had your exam on this stuff. And you did fairly well on it. So, meaning the take-home stuff that you've done for me. If you do that, and you do that, also the practical stuff went really well. What's going on there? So let's do this. Our capillary bed now has the ability for interstitial fluid to increase its pressure to now push into this system here. And all of those are going to be collected to now travel back towards the actual cisterna chyli. Now, I just got rid of that cartoon. Part of it was because I wanted to, but part of it is still having trouble with this pen, and I'll figure it out eventually, but right now. Let's do this. So the next part of the question is, are there larger structures that can make up the lymphatic system? And I'm going to go back to drawing another human. I'm going to make it. There we go. We're going to go like this again. Yay. We're going to go like this. And what this question is asking is a slight modification of the previous one. Is, do you understand the function of the thymus? Do you understand the function of the spleen? And what is the role of bone marrow? Okay, obviously bone marrow is going to be the place where we make white blood cells. Okay, as well as red blood cells, but we're not interested in red blood cells in this chapter. Okay? Having said that, we can now answer what types of cells will you find inside the lymphatic system. So I'm just going to list them. Enough playing around with pens and stuff like that. So let's just go back there, see what happens when I start typing. Okay? Oh, look, I can draw with this as well. That's scary. Anyway, just get out of there. And let's just list these. Okay, because remember on your practicals, for those people who have taken the class with me or have taken the lab practical with me, you know fine well I had you draw all of these. So you know you want all of the white blood cells, you want neutrophils, you want lymphocytes, you want macrophages, so lymphocytes, macrophages. You want eosinophils. And you want basophils. And this is the percent population inside of the white blood cells you find inside the lymphatic system. Also inside the bloodstream, right? So it's neutrophils, highest concentration, lymphocytes, macrophages, eosinophils, and basophils. If we're going to limit it to what's inside the lymphatic system, we want to focus primarily on, let's go like that, we want to focus primarily on lymphocytes and macrophages, right? B-U-I. And that's because these guys, the lymphocytes, are going to be of two types. We have T cells and we have B cells. Whoops. All right, T cells come in two flavors, CD4 and CD8, also known as helper and cytotoxic. Okay, B cells will become activated to become plasma cells, and these secrete antibody, anti-B-O-D-Y. And for the purpose of your final exam, you have to remember that there are 5 different types of antibodies, right? IgG through IgE. Right, and you want to make sure you understand what the different types are, meaning who's the most prevalent, where would you find it, what type of immunization does it protect you against. Okay, but this answers pretty much this first question. We've taken care of making sure you now understand that we're going to get into how do you activate each of these types of cells, what are they actually going to be doing. And notice I'm focusing on the lymphatic system. So that means what we're talking about is if I drew that human being again, we're paying attention to the responsibilities that happen inside that lymphatic system. And for the purpose of clarity when you're studying, remember we're doing a vast majority of the work, we were looking at lymph nodes. Lymph nodes, but remember you also have in mucosal tissue lymph nodules. And the difference, of course, is one has connective tissue surrounding it and the other does not. The lymph nodes have connective tissue. Okay. Next, what are the mechanisms of signaling between cells that inhabit the lymphatic system? You might describe how cytokines work by focusing on the activation of both B cells and T cells. T cells and B cells as an example. You might also describe how the thymus is constructed and how the thymus communicates with cells residing in it. So if I was going to draw something here, it would be that figure inside your textbook that looks something like this. Let's go green, first of all. Green. We're going to draw that big rectangle. All right, there's a couple things I wanted you to pay attention to. You had bone marrow out here, BM. Right, that bone marrow gives you T cells and B cells. T cells will now enter in, so we actually have CD8. I'm going to make sure I go slowly. And CD4. Great. And then the other side of the cartoon over here were those B cells, right? They came in, they're just hanging out, doing their own thing. They've got their own receptor on their surface, Notice I draw the receptor a specific way. And what's going to happen, of course, is something, an antigen-presenting cell, APC, will activate the CD4 cell. And because the CD8 cell has already seen an antigen, okay, the CD4 cell can now co-stimulate it. That's what this question is asking you. What is the interleukin that is required for this? But it gives you the opportunity to review the fact that CD8 activation should not be able to happen unless CD4 cells have been activated, and they're going to make IL-2. Once CD4 cells are made, they make that IL-2, obviously, but they also make larger populations of themselves, so meaning those helper T cells are now clonally expanded, and therefore you've made clonally expanded volumes of IL-2. Same thing's going to happen over here. Should that CD8 cell actually become activated, it will make clonal populations of itself. Yay! It's now going to start running around your body, doing all sorts of horrible things to cells that actually have the antigen that activated it. Okay? And in the end, it's going to keep some memory cells. Remember, it's going to have to remember. R-E-M-E-M-B-E-R. Remember the 5th of November, right? And here's what I need you to do. I need you to remember that this can actually exit the lymphatic system. CD4 cells, we don't draw them coming out of there because they do most of their work inside, say, the lymph node or a lymph nodule or something like that. B cells, should they become activated, and they can, but don't necessarily have to be activated by our CD4 cell, they too will become clonally expanded. And you'll notice how I'm going to change the nucleus here. The cell becomes essentially all just nucleus. And what's going to do is it's going to now secrete that receptor that was on the cell surface. Let me draw that for you. Oops, here we go. We're going to lose the picture. Let's see how we can save ourselves here. I don't know what's happening when this happens, so just bear with me, okay? I'm just going to try and draw an IgG receptor here. Okay, but I also want you to see that now this leaves. And if I could get this image back, let's see if I can get the zoom to go back to where it's supposed to go. It won't, so I apologize. The point is, of course, now you actually have cell-mediated, which are the CD8 cells out here. I'll put those like that. CD8. So that would be a cell-mediated response. And now you also have an antibody protein-mediated response. Did everybody hear that? The difference for specific immune responses. Okay? Every time I think things are getting better, something like that happens. It's sort of frustrating. But I've answered now that part of the question. But let's take just a quick look at the thymus, right? So the thymus, remember, it sits anterior to the, it's just slightly superior to the heart, okay? Anterior to the heart, inferior to the, or deep to the sternum. And it has within it T cells that are immature. So we're going to put a little arrow there like that. T cells travel here from the bone marrow to mature. Okay. Once they've matured from here, and they know this because the thymus is communicating with the cells using thymopoietin, okay, and other thymic hormones that regulate the time, the duration they spend here. Then they move to, say, a lymph nodule or a lymph node, okay? So that's what I'm actually asking in the second part of this question here. Let's get rid of that. Bam. Let's move to the next one. Okay. What are the mechanisms for an immune response from in total from beginning to end? Okay, and this is actually a fairly complex question. So what I'm going to do is I'm going to answer it in terms of brackets. What do I mean from beginning to end? I mean from initial presentation of antigen to antigen presenting cell. Okay? Presentation. I don't understand. Presentation, antigen, presenting cell. So this could be our macrophage, right? Let's go just put macrophage here. Right? And now, if I'm asking you what can happen from the outside of the cell, you know then that means that skin, sweat, etc. is nonspecific. Nonspecific response. That should something get through that, then you actually have a continued nonspecific response with the macrophage ingesting whatever actually got into the body. And now it presents this. Presents at cell surface. Okay? Makes sense? Okay. That's what it looked like from the outside. So your skin's actually doing most of the work here. Sweat and the materials that are found inside sweat will do the next layer. Should something get through, macrophages and all of your clotting mechanisms obviously are going to stop some amount of materials that get into the body. Now, can I break this down to what I'd see at the tissue level? Well, what I'm asking you here is, let's go like that. What I'm asking you here is what's happening at the lymph node. Lymphnode. And at this time, what I'd probably do is draw a lymph node for you, right? And I know you can all draw lymph nodes because you just did this inside your lab practical, right? Where you're going to have, you know what I'm going to do? I'm going to do this. Screw it. I'm going to do this. I'm going to go like that. And I'm going to go, lymph flows through an afferent structure into the subcapsular sinus, right? Where you can then move into the trabecular sinuses. So what you're seeing, of course, is you're creating these areas where we're going to decorate with cells. What are the cells that are inside these cortical, so this is the germinal center here. What are the cells that are surrounding it? And then as we move towards the efferent outflow, right, and that efferent outflow means something, right? It means that an area of lower pressure down here, so there has to be some sort of sinus, okay? And it's called the medullary sinus. What cells are found here? Okay, so that's what I mean when you're looking at the tissue. How is this different from, say, a lymph nodule versus, say, a lymph node? Okay, and where else, do you find anything else like this inside the body? And if the answer is no, I mean, that might mean something. It means Mother Nature has figured out a way to concentrate immunosurveillance to specific structures. Now, what's actually happening at the cellular level? Here's where things get a bit tricky, right? I want to switch colors here. Because at the cellular level, we need to think about, say, complement. So let's put just complement fixation here. We want to think about what's happening as a specific response. So we'll be T lymphocytes, right? And, you know, you have the LYMP, right? LYM lymphocytes. And then, so that would be specific immune responses. And then over here you have non-specific, okay? And that could be anything from skin through, say, macrophages, okay? And what I want to make sure you pay attention to here is you should be able to discuss what are the different pathways for complement fixation. You know, why is it being activated? Well, it's because there's an increase in the number of bacteria that are present. That's why, all right? It's really quite simple. T cells become activated once the antigen that can activate that unique T cell, or B cell, I should put there, you know. T lymphocytes, I should put B lymphocytes here as well, right? LYMP. I know this is so hack, I'm so sorry. And then everything's always being surveyed just randomly. So macrophages can find something, they'll activate it. But how all these work together, that is pretty much what you're spending most of your time thinking about. Okay. So let's go like this. Let's go like that. Let's move on to the next question. What is phagocytosis and what are the steps involved? Okay, so we just mentioned the fact that you can actually have non-specific moving around of materials inside the body, right? So remember, here's our famous macrophage, right? It's going around, And what I need you to remember, and what you need to remember, not just I need you to remember, is that these are ingesting materials. They're turning them into a vesicle. That vesicle is now going to meet the major histocompatibility complex structures. And because it's been ingested from the outside, that's exogenous. So we would use MHC2 here. So let me put that right there. MHC2. Now, in a not so distant world, macrophages can get infected too. So let's imagine a virus comes in and infects a macrophage. Doink. Okay. And now it is endogenous. And in the case of endogenous presentation, then we would use MHC1 to present whatever that material is on the surface of the cell. So we could put MHC1. Now make sure you pay attention to the fact that I'm stressing that you have to understand that both of these are going to activate T cells and B cells differently. And even more precisely, T cells, meaning T helper cells, and cytotoxic T cells. So I'm going to put an 8 here to help you understand MHC1, particular to cytotoxic T cells, MHC2 here for helper T cells, right? Where CD4 stands for helper T cell. So what's the big deal? Why is phagocytosis so important then? Well, remember, whatever it is you ingest, let's make this a whole bacteria here. Bacteria can be infected too. Okay, whatever is ingested, though, is going to be now presented on the surface of this antigen-presenting cell. So phagocytosis is important for making sure that antigen presentation occurs, APC. And that happens throughout the body. It will travel into a lymph node or something like that to make sure that you're protected. So this is a pretty good place to stop. Notice I didn't go through like names of steps, endocytosis, fusion of blah, blah, blah. Take a look at it. That's where the definitions are going to be found. But why is Mother Nature doing it? That I think is actually in many ways more important. Okay.